Hello again. Right, we're just going to take stock. The last couple of lessons you'll be doing the required practical that goes with selection, and I'll say more about that in the other video when I do my explain everything type thing. I thought it's worth going through what we've done and where we're going next. So we're still in topic 4, and we'll finish topic 4 in the next couple of weeks, and then we start a year 13 topic like we always do, because the biology syllabus requires it, as in to get through it in time, even though we have extra teaching time next year because we'll see you 5 times a week instead of 4. Anyway, we've been doing species and taxonomy. Okay, there's no skills, as you can see in the syllabus, so we don't have to worry about that. We've been doing about courtship behaviour. I think that was the last thing we did in terms of content. Then we looked at the phylogenetic classification system, those like spidery type diagrams that, those are non-overlapping hierarchy. We know the king, prawn, curry, or fat, greasy sausages. Have I mentioned that domain comes first? Probably not, but domain comes first, which would be either eukaryota or prokaryota or archaea, but you don't need to know any examples anyway. Each species has a binomial, so we've mentioned that it's genus and species name, and the recall of the different taxonomic systems will not be required. So you just need to know the order of domain, king, prawn, curry, or fat, greasy sausages. And then crucially, it says at the bottom, students should be able to appreciate that advances in immunology and genome sequencing help to clarify evolutionary relationships between organisms. So we're going to look at immunology again today to see how we can use immunology to know how related different species are. So that's 3.4.5. We're going to come back to 3.4.6 because it just fits in better to do 3.4.7, which is the last bit of topic 4. So we will be going back to 3.4.6, but it makes sense to kind of do this now. As you can see, there's loads of skills here. And all of this about sampling within a population, that's what we're going to do in a more thorough way once we finish topic 4. So there's kind of sampling light in year 12, and then there's full-blown sampling in year 13. And this is meant for that if we're doing AS exams, we do this, but we don't do AS exams anymore. In fact, most places don't do AS exams, and you couldn't have done them anyway this year. So we save time by covering this when we do it properly anyway in the year 13 work, but we get onto that in a couple of weeks anyway. Then talks about using calculators to calculate mean values, really simple, and about interpreting standard deviation. If you're wobbly on maths, we are coming on to quite a bit of maths. But at the bottom here it says, where's my pointer gone? There it is. Students will not be required to calculate standard deviations in written papers. So you don't need to worry about ever calculating it. Interpreting it is the important bit. Anyway, back to the content. It says genetic diversity within or between species can be made by comparing. We can compare the frequency of measurable or observable characteristics. That's like, who can roll their tongue. I'm going to make some data later, I think, on blood groups in different populations, because that's quite interesting. There's the ABO system, rhesus positive, rhesus negative, and all that. So we'll have a look at some data probably on Friday based on that. We can look at the base sequence of the DNA or the base sequence of mRNA. These are really modern ways of doing it, and we couldn't always do it this way. So we're going to kind of look kind of historically. How can I compare? I've got these two species. How different are they? Well, I can kind of measure them by, now you can just do a genetic. You can just take some cells and get the sequence of DNA and compare them. But what did we used to do? And it brings in immunology. Or I can look at the amino acid sequence of proteins. So it's all about comparing organisms by really comparing the DNA either directly or indirectly by looking at the mRNA, or the amino acid sequence of a protein, or what we're going to do today, the shape of a protein. So we'll skip this first bullet point, but we'll get to it in a bit. But on this second bullet point, it says appreciate that gene technologies cause a change in the methods of investigating genetic diversity. Excuse me, a little burp. Inferring DNA differences from measurable or observable characteristics has been replaced by direct investigation of DNA sequencing. So we're going to go back to look at the old method so that you can appreciate why the new method is better, if that makes sense. Okay, most of this relies on your knowledge of immunology. So I'm going to zip through this, then I'm going to go through it again on that explain everything, where I'll be writing on a worksheet to take you through. Okay, so yeah, we're going to do the immunological comparison of proteins. This isn't mentioned directly on the syllabus, but it has said about using immunology in one bit of the syllabus, and we need to be aware of kind of older methods. So they can never ask a question saying 6 marks to describe immunological comparison of proteins, but they can give you information about this and ask you to come up with conclusions. So we're going to look at the protein albumin. You'll have heard of albumin, hopefully. It's the white of an egg. It's a globular protein. It's a soluble globular protein, like most globular. In fact, I can't think of any non-soluble globular proteins. And it's the same protein that's found in blood. Okay, so if you've been doing on the other side of the course about tissue fluid formation, it's the plasma protein that's in our blood, and it creates a more negative water potential because it's such a large protein. It isn't filtered out by the arterial end in the capillary. It stays in the capillary, and therefore it causes water to move back by osmosis. Okay, so yeah, it's the plasma protein. And in terms of its shape doesn't really matter. It just needs to be big and soluble. Okay, and this question here says, what determines the shape of the protein? Remember when we did this, we talked about sequence shape, where the sequence of bases in the DNA called for a certain sequence of amino acids to be joined, and therefore those amino acids have R groups that cause a specific tertiary structure in the protein because they form hydrogen bonds, etc., to pull it into a complex 3D shape. Okay, so sequence shape, and we did a bit about that when we did about mutation. And if we ever get back into school, that's what I've planned for half of your session to really. We'll be recapping kind of mutation, meiosis, and getting into natural selection in like half of your 3-hour session if we ever get back in. But hopefully that'll be confirmed soon. So yeah, so because all you have to be is a large protein, a mutation doesn't really affect it. Yeah, if it was an enzyme, a change to the sequence of bases called for an enzyme will cause the enzyme to have a different tertiary structure. The active site will change. It won't work as an enzyme, and therefore, in terms of the advantage to the organism, it's probably going to die because it lacks a vital enzyme. Okay, but you can mess about with albumin. It doesn't really matter as long as it stays big and soluble. It doesn't matter. So it's highly variable between different animals. That's like a possible suggest question. Suggest why they used albumin rather than amylase. Yeah, why they compared them. My amylase is the same as dog amylase, is the same as chimpanzee amylase. It has to be the same shape because it works on the same substrate, whereas albumin can vary because it just needs to be big and soluble. Right, brace yourself for my PowerPoint artwork in a minute. It gets better than this. So this diagram's showing pretend albumin. Okay, so on the previous slide, I've kind of showed albumin as this blob, and I've put shapes in the outside of it to show that it's got a shape. And human, dog, and ape are shown here. It's a generic ape. It could be any ape. Let's say it's a chimpanzee. And you can see that the shape of the human and the dog, they vary. However, they both have this diamond shape in common. Okay, whereas the ape has got quite a lot in common. Crucially, we can't see proteins. Microscopes are not powerful enough. Yeah, so we need to develop a way of measuring the shape of a protein without seeing the protein. Okay, and this is where we use our knowledge of immunology. So now we get to my brilliant artwork. That's a human, and we take a blood sample. I don't know why I've taken it out of his neck. Maybe I've gone into his jugular vein. How to Kill a Circus. Go for the jugular. Little light-hearted relief there from blood being taken out of a human. Anyway, so we take blood out because that's where the protein is, and then we purify it so I get just the plasma proteins. So how can I get rid of all the red blood cells and white blood cells and platelets? Yeah, quickly. Yeah. Well, what technique do we know for getting rid of large things out of a liquid sample? Exactly. We spin it in a centrifuge. Therefore, that forms a pellet. I then pour off the plasma, and that will contain the plasma proteins, and we'd call that the supernatant. But in this case, we just spin it quickly, get rid of the cells, and then I've got my my plasma. So I didn't homogenize it or anything like that. I just did it. It's a fast way of getting rid of the cells. If you use a filter, the cells will just block the filter. It's really hard to kind of filter when you're on a very small scale of cells. So yeah, we just spin it in a centrifuge. So I've got now, here's my little test tube full of human plasma that contains plasma proteins, albumin, and other ones as well. Next, we use, this is where people get confused. This is just an animal that's very dissimilar, but works in a similar way to humans. So I'm going to compare humans and dog, and humans and chimpanzee. Okay. Therefore, I think, well, rabbits are very dissimilar. I'm going to use a rabbit. I could use a rat as well, but I'm good at drawing a rabbit on PowerPoint. So it's almost like giving it a vaccination here. I inject a small amount of the human plasma protein into the rabbit. Yeah, it's not nice for the rabbit, but at least it's safe from foxes and it's fed well. It says, describe the immune response that results in antibody production. And you can see how they can mix questions around, kind of, and it's what they like to do is build, like I can get a bit of topic 2 and a bit of topic 4 in one question. They love doing that. So yeah, describe the immune response. What's the first cell? So it's got some foreign protein in its body. It's very different to its own protein, and therefore it will elicit an immune response. So the first cell there are the phagocytes. Yeah, they perform what process? That engulfing, phagocytosis, and then they change into antigen-presenting cells. Yeah, what part of the body of the rabbit do they then go to find the lymphocytes? They go to the lymph nodes. That's 4 marks, well, possibly not 4 marks. And in the lymph nodes, they find a B cell and a T helper cell that have complementary receptors. Okay, the T helper cell activates the B cell using cytokines. Don't know why I'm saying hold up 7, but I am doing. And therefore that B cell gets activated, which then clones by mitosis into plasma cells. The plasma cells then secrete antibodies against the human protein. Okay, so could you have gone through all that detail? I've not talked about just T cells because that's the cell-mediated response. I've described the humoral response, but I referred to T helper cells because they are the ones that cause the activation of the B cell. Okay. So yeah, I let the rabbit do that, and then once it's gone through its immune response and there's loads of antibodies in its blood, I then take a small blood sample. Again, it's not that much in size. It's a few mil would be enough, a few centimeters cubed. So we take a little blood sample from the rabbit because in there is going to be the antibodies. So there's the rabbit's blood sample, and these are diagrammatic versions of the antibodies. I know they really have 2 binding sites. I'm just showing them with 1 binding site. So yeah, because the human albumin's got 4 regions, I'm just making this up. It's got 4 parts of its molecule that are recognizable as being foreign. Then the rabbit, that's very different, has had an immune response against every single region of the protein. So these now are called anti-human albumin antibodies. So they will bind to the antigens on human albumin. Bit of a mouthful. Okay, so I've made the antibodies against human protein to say it faster. So now I can do the sip. I know I peeked on my dog. Look at that. So I'm going to compare a human to a dog. Okay, so I take a little blood sample out of the dog. Again, doesn't harm it, apart from a little injection. Well, not injection, a little removal. And I'll have to take the plasma proteins out. So this is dog plasma protein, and we add the antibodies that I've gained from the rabbit that are anti-human. Okay, and then all we measure is the amount of a precipitate. Remember when we did about how antibodies work, about agglutination or precipitation of soluble toxins? Because antibodies have got two binding sites, they will bind onto one protein and another protein, and they cause it to go from soluble to insoluble. Again, I'll show you more on the sheet when we go through this again, which will give you another kind of run through, because it is complicated. Is this again all this detail you don't need to know, but they can build a question from it by telling you this information. So there's dog albumin from before, and you can see that only one antibody will bind to it. Yeah, it hasn't got a lumpy antigen, it hasn't got a square antigen, and it hasn't got a triangular antigen. Yeah, the only one it has is the one that matches this one here. So it's only got one antibody that's bonded, so it can only do a small amount of agglutination. Okay. However, if I couldn't draw an ape, if I could draw, ignore that. Yeah, I can't draw an ape. So this is the ape's protein. The anti-human antibodies here would bind to 3 of them. That would cause a lot of agglutination. If I put human plasma protein, I would have full agglutination. So I can basically infer how similar one animal is to another by seeing how much agglutination takes place, and that would be judged by the volume of a precipitate. It's a bit like when you did Benedict's test. If there was a bit of sugar present, you get a bit of colour change, you get a bit of precipitate forming, and you can actually filter that precipitate off and dry it and weigh it. It's something that we don't do because it's messy and takes time. We just judge the colour. So yeah, the more similar you are to human, the greater the precipitate you will get, and therefore it just gives you a way of measuring it without ever seeing a protein. I can just take a blood sample. I have to use that second animal, that rabbit or a rat, something very dissimilar to the animals that I'm comparing me to, but I can get a measure of how related we are. So yeah, apes like chimpanzees are more related to humans because there's greater agglutination, and I'll go through that agglutination in the next video. So sorry that's been a lot of me talking, but hopefully it's made sense. And let's have a look at that dog again, because that took me, how long did that take? About half an hour? Years ago. But yeah, it took some time.